Welcome, everybody. We're very excited to have you here. We have over 200 attendees registered from over 10 countries around the world. So in addition to connecting with the presenters, we hope you have the opportunity to connect with each other. As we begin, I would like to acknowledge that I'm joining this event from the ancestral, traditional, and unceded territory of the Coast Salish peoples, including the territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. I'd also like to acknowledge that others are joining us from other traditional and unceded Indigenous territories across this beautiful province, country, and around the world. Thank you for joining us. We're very excited to be here to host this event virtually. Over the last few years during the pandemic, Life Sciences BC and our amazing life sciences community have fortunately embraced virtual events. We're grateful that we've been able to continue to host webinars and bring our ecosystem together virtually. This event, of course, is a virtual event in advance of one of our first 3D or in-person conferences in the last 2.5 years. So the attendees that are presenting today will be at Bio next week, in addition to ourselves and approximately 15 or 20 other BC-based companies that we hope you'll have the opportunity to connect with. At the end of the presentation, we'll have a QR code on the screen, which you can click on to get information about all the companies that will be attending. For those of you that do not know, Life Sciences BC is a not-for-profit organization. Our members consist of academia, research institutions, centers of excellence, and funding agencies, emerging small and medium-sized and growing digital health, medtech, therapeutic companies, and those organizations that support the life sciences sector broadly. We're funded through membership and the generous contributions of sponsors and partners. And Bio is one of those events that has had that generosity. We would like to thank our international networking reception presenting sponsor, Ferris, our BC Partnering Room events supporter, Novature, and our BC San Diego supporters of our delegation generally, the BC government and Invest Vancouver. We hope that you'll be able to connect with them while you're in Bio next week. We're very excited about today's event, the BC Showcase for Bio San Diego. We look forward to showcasing our innovation from our companies and our ecosystem broadly while we're there, and hope to connect with you. I'd like to start off by providing a brief overview of the life sciences sector, which I believe the slide is going to carry on. So a little bit about our sector. We are home to the third largest life sciences community in Canada, and we are the fastest growing life sciences sector in Canada. We have the largest employer in stem cell technologies. That is the largest biotech in Canada. And we have numerous companies that are over the 1 billion market cap mark. Abcelera, Sierra Oncology, Aurinia, Xenon, Chinook, and many more racing up to that milestone. Based off of data from a couple of years ago, the life sciences sector is $5.4 billion in revenue, a 1.6 billion GDP contribution, 500 million in exports, and our companies are growing at 5%. We suspect that that is actually going to, as we update this data this year, show that life sciences is the fastest growing sector in British Columbia. We also employ almost 20,000 employees. There's 20,000 students studying in STEM-related fields, which is one of the fastest growing areas of academic schooling in our province. And we've seen $2 billion of capital come into the province in 2019 and 20. Some things as you think about the BC biotech sector, we are very strong in antibodies. We've got 20 years of antibody innovation and numerous companies that operate in that space in our sector. Regenerative and cellular medicine, nanomedicine, lipid nanoparticles, oncology, genomic-based precision medicine, and gene therapies. We have a very strong medtech sector in medical device design, prosthesis, and 3D printing. Digital health is an area of great strength for us, and creating solutions for human health, which combine physics, biology, chemistry into a human engineered solution. As you listen to our presenters and learn more about our sector, you will see that there are numerous innovations that bring together different scientific disciplines, which is where we have deep, deep strength over numerous decades, and as you look ahead at what's coming. Life science is an important point of connection between human health, agriculture, food science, and clean tech. And we continue to see our innovation that comes from the life sciences space having strong applicability into these areas. We also are a hotbed of talent, which we're very blessed with people continuing to come to BC to work in our sector. We actually hosted an event annually, which is referred to as Career Connect Day, where we saw 800 people attend, all interested in pursuing careers in BC. So super proud of our BC ecosystem. Another thing is that we learned during COVID that when necessity strikes, the BC ecosystem can really rise to the moment. We acted fast, the importance of science-grounded innovation, global collaboration and partnerships, and everyone working together with research, industry, government, and regulators coming together. BC Innovation is actually the delivery mechanism of the Pfizer vaccine, which is made by Acute, came out of BC. We saw the first therapeutic come out of BC Innovation with Accelera partnering with Lilly, and numerous companies in BC that were operating in the testing space, the monitoring space, the apps, the digital health tools. So BC really was able to rise to the moment, both to the benefit of BC and Canada, but also the globe. And these are the list of all of the companies from BC that are going to have a presence at BIO. About eight or nine of them you're going to hear from today. And again, there will be a QR code with a link to a booklet where you can learn more about the companies and how to connect with them and what their objectives are at BIO next week. With that, I would like to introduce our honorary speaker, the Honorable George Chow, Minister of State for Trade, Ministry of Jobs, Education and Economic Recovery, sorry, not education, Economic Recovery and Innovation, who will share a few words. George was elected MLA for Vancouver Fraserview in 2017. Prior to that, he served 2 terms on Vancouver City Council from 2005 to 2011. He is the Minister of State for Trade. Born in China, he completed primary school in Hong Kong and settled in Vancouver in 1965. George grew up in downtown East Side and attended Britannia and Gladstone secondary schools. He graduated from UBC in mechanical engineering in 1975 and had a 30-year career with BC Hydro. He's a member of the Association of Professional Engineers and Geoscientists of BC and a fellow of the Engineers of Canada. He's also served on many community organizations as a volunteer, including the Urban Spirit Foundation, Vancouver Public Library Board, Chinese Cultural Center of Greater Vancouver, Boys and Girls Clubs, Chinese Benevolent Association of Vancouver, Vancouver Chinatown Merchants Association, and many more. He's actively involved in the community and is a key leader within the BC government. And with that, I will turn it over to Minister Chow.
Okay, I just can you hear me?
Yep.
Okay, thanks. Thank you, Wendy. And hello, everyone. I'm George Chow, and I'm joining you online from Vancouver on the traditional unceded territory of the Coast Salish people. So as Minister of State and on behalf of the government of British Columbia, I want to thank you for inviting me here to speak about our thriving life sciences sector in British Columbia, Canada. You know, when I was in high school, biology was not my favorite subject, and that's why I went into mechanical engineering. But 40 years later, this is the sector that's leading us in British Columbia. So I'm very happy to be speaking to you today. And of course, I want to acknowledge some of the companies that make BC a global leader in this field. Dr. Mohsen Abari, he's the co-founder and chief scientific officer of 4M Biotech. Dr. Gregory Block, Senior Vice President, Corporate Development, Notch Therapeutics, and John Barkley, Vice President, Business Development, ABO Science. Dr. James Halliwell, Chief Executive Officer, Apraxia Pharmaceuticals, and Mr. Handel Kim, founder and CEO of Variational Ai, and Robin Sharington, Executive Vice President, Strategy and Innovation, Xenon Pharmaceuticals, and Julie Rezler, Vice President, Corporate Development of Primary Peptides, and Karim Laji, who's the CEO of Microbiomes, and Yvonne Martin, Executive Director, Providence Health Innovation Research and Engagement of Providence Research. So these companies I've listed are just a few of the examples why BC has become home to the fastest growing life science sector in BC, in Canada. As Wendy pointed out, we are the third largest in Canada and it's growing. So building this sector in BC, to be a world leader has never been as important as it is today. And of course, in the past 2 years, we have been seeing extremely challenging events that are changing lives, changing economy, and changing our future. And the pandemic, along with the increasing extreme weather events, as well as the war in Ukraine, all had impact on the global economy. And this tells us and has highlighted for our need to develop biomanufacturing capacity right here at home in BC. And we have inspiring successes from our own life sciences sector. And think about this, and I think Wendy mentioned it, I'm very proud that virtually every COVID-19 vaccine that reached late stage development in 2020 used components that were initiated, manufactured, or developed by British Columbian scientists. It's quite an accomplishment, and as I said, I'm very proud of our life sciences sector. As you may know, we recently launched our Stronger BC Economic Plan, and we are moving forward with a clear direction to achieve inclusive and clean growth. Our plan is a made in BC new approach to economic policies. To help us meet our goals, we are taking immediate action, and that's why we'll be launching a life sciences and biomanufacturing strategy to position our province as a world leader in life sciences by nurturing new talents, developing new lab space, utilizing our world-class post-secondary institution, and creating jobs right here in British Columbia. And that's not all. This spring, we announced $195 million in funding for Michael Smith Health Research BC and Genome BC as well, which is a significant step towards anchoring British Columbia as a world leader and developer of new talent, research capacity, and life-changing innovation. Technology and innovation are strong economic drivers in British Columbia. This sector is a top provincial performer, like Wendy said, with over 2,000 companies in British Columbia. Through InnovBC, our crown agency in charge of developing innovation within BC, our government is providing support to businesses, entrepreneurs, and non-profit across the province to bring new products and ideas to market, adopt leading-edge technologies, and to secure talent while opening doors for economic opportunity for Indigenous people and traditionally underrepresented groups. We continue to invest significantly where it matters most, and in people, that's where it matters most, by making a generational commitment to continue to develop talent here in British Columbia, so people have the skills needed for the jobs of tomorrow, especially in biomanufacturing and the life sciences sector. So British Columbia is committed to true, lasting, and meaningful reconciliation with our Indigenous peoples, including through our economic development. We foster a rich multicultural society and value the contribution of everyone who makes to our sustainable, innovative, and inclusive economy. This is a key pillar of our Stronger BC economic plan. We prioritize and support low carbon initiative and green business practices, Through our Clean BC plan, we are ensuring BC leads as a supply of high demand, low carbon goods and services. We're also supporting our business with the development of our environmental, social, and governance strategy and ESG Center of Excellence. Our high standards for environmental protection, social support, and democratic governance are an important and growing economic advantage. These standards are becoming increasingly important to investors and partners around the world. British Columbia is bursting with innovative entrepreneurs who have export-ready products that tell the story of our rich, diverse, and fascinating province. So please reach out to the business we have showcased. Let us know what you're looking for. We have many business opportunities to explore together. I look forward to hearing about your new collaboration and partnership with BC businesses. Thanks again for inviting me to speak at this important conference. It's always a great day when we work together and collaborate. Thank you.
Thank you very much, Minister Chow. You're a great supporter of our sector, and we really appreciate it. And thank you for taking the time to deliver opening remarks and kick off this webinar. With that, I am going to introduce our first speaker, Dr. Mohsen Kabari, co-founder and chief scientific officer at 4M Biotech, who will be speaking about his company and his goals at Bio.
Well, thank you very much, Wendy. Can everyone hear me?
Yep.
Wonderful, wonderful. It's a great pleasure to be here. Thank you very much for the invitation and also for giving me this opportunity to showcase a technology that we've been developing over the past few years in my company, 4M Biotech. I'm the CSO and co-founder of 4M Biotech. I'm also wearing another hat, which is a professorship hat. I'm an associate professor at University of Victoria. So 4M Biotech is a University of Victoria spin-off, which was founded in 2018 with the aim of developing technologies in the digital health sector. So this is our website. If you're interested to know more about what we do, feel free to check us out. But one of the areas that we are very much interested in is wound management. Wound management is a huge, expensive, and growing area. 70 million people around the world are dealing with either traumatic or chronic wounds. And in the US alone, $50 billion are being spent on wound management by the healthcare system. And these numbers are increasing over the next few years due to our inactive lifestyle, obesity, and unhealthy diet, and then also increased age population. Actually, it was estimated that over the next 20 years, Canada's seniors population will grow by 68%. And so you can see that this problem is going to grow over the next few years. And infection is a big problem. In wounds, up to 58% of pressure ulcers, they become infected. Infection can significantly delay wound healing and increase hospitalization stays up to a few months. Infection can also lead to amputation or sepsis in severe cases. Actually, International Diabetes Federation recently announced that every 30 seconds, one leg is lost to amputation due to infected wounds. Despite the magnitude of the problem, wound monitoring has not been changed for centuries. So currently, individuals with wounds need to go and visit a doctor in person. The doctor removes the dressing, looks at the wounds for any sign of pus or excessive exudate, color change, or swelling. They even smell the wound sometimes. It's well known that the grape-like smell is basically an indication of Pseudomonas aeruginosa, which is the most prevalent type of bacteria in wounds. If they're suspicious that the wound is infected, then they take swab samples and then they send the samples to a microbiology lab for a confirmation and then also identification of the type of pathogens or microbes that are present in the wound. So as you can imagine, this process is fairly subjective. It's time consuming. It takes time for the patient to go and visit doctor, wait at the doctor's office, and then also get the results. It takes 24 to 48 hours to get the results from a microbiology lab. The process is invasive. The doctor needs to remove the dressing to be able to access the wound and assess its condition, and removing the dressing will increase the chances, will expose the wound and increases the chances of infection, and requires frequent doctor visits a few times a month to ensure that the wound is healing and then there are no infections. But visiting doctors is not usually always an easy option, especially for certain populations, for example, for individuals with impaired mobility, elderly, or those who have spinal cord injury, or for soldiers in war zones, those who live in remote areas. And then we have all learned during pandemic that in-person visiting doctors in person is not as easy as we thought. So we think that we have a solution to this major problem. Our solution is called Gelderm. Gelderm is a smart dressing that detects infections using embedded color changing sensors. The doctors or the patient can apply the dressing onto the wound and then secure it in the wound using a secondary dressing that has an adhesive layer. The color of the sensors are fairly transparent, but when the wound becomes infected, the color of the sensors shift to a dark green color, which is an indication of infection. We provide the patients with a color code that facilitates detecting infections more objectively. A unique feature of our sensors is that this color change is reversible, which means that if the bacteria or the infection is cleared from the wound, the color will go back to its original transparent color. And that can be used to assess the effectiveness of the treatment strategy that was used by the healthcare system. So we have made tremendous progress since the inception of the idea in my lab in 2015. We created the first prototype in 2017, and then we've performed extensive bench validations and small and large animal validations in our lab, and then also in independent labs. We're looking into performing further performance validations in clinically relevant animal models, which will be a pig, and then also performing safety and performance validations in GLP facilities before we can move on to the next step, which will be in human studies, and then obtaining FDA and Health Canada approvals. We've also developed a mobile application, which is which interfaces with the sensors. It has a machine learning algorithm that helps us to increase the objectivity of the sensor detection, and then also it enables telehealth capability. We are very excited to participate at the Bio International Convention. Our primary goal is networking and making ourselves familiar with the biotech ecosystem in California, in this area. But we are also seeking partnership opportunities for co-development of our technology, and then also we're hoping to meet potential investors who can help us to further develop this technology. With that, I'd like to thank again the organizers for inviting me to this very exciting event. Also, I would like to thank the participants for listening to my talk. If you have any questions, feel free to reach me out using my email, and then also follow me on Twitter. Thank you.
Thank you very much. Great presentation. And we appreciate you taking the time to participate in this and look forward to seeing you next week in person. I would now like to introduce John Barkley, Vice President of Business Development of Abozymes Biomedical, who will be sharing with us the work that is happening there. Abozymes is a preclinical stage company with an innovative platform that enables a world where a person's blood type is no longer the dominant factor. So I will move it over to John now.
Good morning, everyone. Thank you very much to Life Sciences BC for this opportunity. We are Abozymes Biomedical, and we are making blood types obsolete. So there are four main blood groups, A, B, AB, and O, and your blood group is determined by carbohydrate antigens on cell surfaces. These antigens are actually present on all endothelial and epithelial cell surfaces, not just red blood cells. So this is why blood type matters for organ transplants as well. And as you are very well likely know, Group O is the universal donor blood group. So what we want to do at Abozymes is convert all donor blood and all donor organs to O, the universal donor group. So how do we do that? Our technology is actually really straightforward. We make enzymes that convert the blood group antigen to O, the universal donor type. The enzymes are highly specific and highly efficient, and they work on all cell surfaces. Our data and claims can be seen in a 2019 paper in Nature Microbiology concerning the blood conversion of whole blood and packed red blood cells. And more recently, we had a paper published in Science Translational Medicine that demonstrated the ex vivo conversion of intact human lung. Now, that last paper was particularly exciting for us because it resulted in a significant amount of international media attention. So we have blood, we have organs. These are two very distinct market opportunities. So what we are grappling is determining, well, what's the better first market opportunity? So it's called blood group. So our mind tends to go to converting donor blood first. And the somewhat obvious application of this technology is increasing the amount of universal donor blood, O negative, which is used in trauma scenarios in which there's insufficient time to determine the patient's blood type. Furthermore, the supply of O negative blood in terms of like national inventory is almost always at the critical level. However, for a commercially viable product, we still require a filter to remove the enzymes prior to transfusion, so it fits seamlessly into the blood processing operations. Now, fortunately, our academic collaborators at UBC have a $5 million grant from the US Air Force and able to develop this technology. So if we flip our lens to organs now, globally, there are about 150,000 organ transplants per year. About 40,000 of those happen in the US. And last year, there were 25,000 kidney transplants per year. However, any given time, there's as many as 100,000 patients on the kidney transplant waiting list in the US. A kidney transplant leads to better health outcomes and is much more cost effective over dialysis. That you have things like the Advancing American Kidney Health Initiative, which among other objectives seeks to have 50,000 kidney transplants per year by 2030. Now, these images, show the dire situation of those on hemodialysis and on the kidney transplant waiting list. That you see these sort of public pleas for a live donor that matches blood group. Now, why blood group? Well, blood group, the time on the wait list varies by blood group, and B type and O type patients wait the longest, and they're also overrepresented on the waiting list. So these blood groups are also overrepresented in certain ethnic populations, so it exacerbates the health inequities that they already experience. So if you think about converting kidneys to O, there's a number of benefits, and it sort of starts with increasing matching opportunities. So I split this out into living donors, deceased donors, and procurements and discards. For living donors, we can increase the living donor transplant rates. And one way to look at that is through kidney paired donation programs. Now, within these programs, you have a a high supply of A donors. However, the demand isn't for O patients. So what ends up happening is that A donors are stuck on the waiting list. Now, if we go to deceased donors, we can prove the equity of donor organ allocation, as I sort of described on the previous slide. And also, there's also a procurement and discard issue. What constitutes a high quality organ suitable for transplantation is highly subjective, and it's influenced by blood type. As one transplant surgeon said to me, a marginal A might turn to be a pretty good O. The purpose of this slide is to show that with our technology, there can be profound impacts to organ procurement and allocations. Therefore, they have been very much a focus, or the organizations involved in this are very much a focus of our customer discovery. And also another important aspect is that the way our technology will be delivered, the costs can be bundled into organ acquisition costs, which are favorable from a reimbursement perspective in the US. Which begs the question, how is our technology developed? It's pretty straightforward. So when a kidney is harvested from the donor, it's placed into a commercially available organ perfusion device in which it's maintained, or it's perfused with a solution that contains our enzymes. The enzymes are rinsed out from the organ prior to transplantations, which has also the benefit of a medical device regulatory pathway for us. Now, speaking of which, we have a Q-Sub meeting with the US FDA on July 28th to clarify our regulatory path, specifically where we land between a Class 2 510K and a full PMA. And we are looking to submit our IDE package in the Q1 or Q2 2023. Here's a snapshot of for our blood transfusion product. It's a little staggered back to develop the filter, and also we're in the process of developing the B to O conversion enzymes analogous to our A to O conversion enzyme. And this is all gearing towards our initiating our Series A financing later this year. Here's a snapshot of our team. John Coleman is our CEO. His last successful venture was he was a co-founder and COO of Anandia Laboratories, which he successfully exited in 2018. Dave Matthews is a very experienced biotech executive, being part of raised over $500 million, 5 successful IPOs. Dr. Peter Rothfeld discovered the enzymes, and we've also brought on Dr. Christopher Nwan, who is a kidney transplant surgeon at Vancouver General Hospital, which is a high volume kidney transplant center, as our chief medical officer. In brief, we've developed an enzyme technology, converts blood group antigen to universal donor group. We're focusing on kidney transplant first, but we're also expanding to other solid organ types in blood. Medical device regulatory pathway, so it's an efficient development path to a very large global market opportunity. Now, specifically for bio, this is about having conversations in advance of our Series A financing with potential investors. But we also have meetings scheduled with potential partners on both blood and the organ side, and as well as some other niche applications of our technology, which I haven't gotten into in this presentation. Anyway, that's it. I'm happy to speak with anyone. My email is below. You can reach out to me on also on the partnering platform. So we still have a few meeting slots left. So thank you very much for your time.
Great. Thank you very much, John, and a great presentation as always. And it's now my pleasure to welcome Dr. Gregory Block, Senior Vice President, Corporate Development, Notch Therapeutics. Notch Therapeutics is an early-stage biotech company working to maximize the benefit of cell therapies through a proprietary T-cell production platform, which combines sophisticated product design with commercial compatible processes to enhance patient outcomes. Over to you, Dr. Block.
Thanks, Ryan. Can you hear me okay?
We can hear you great, and your slides look perfect.
Excellent. You guys are covering my slides, so this is going to be fun. It's a pleasure to be with you guys today. Thank you very much, Life Sciences VC, for the invitation. Today, I'm going to talk to you about how Notch Therapeutics is engineering therapeutic T cells from induced pluripotent cells. why we think we can be the best in the industry to do that, and the impact that that's going to have on medicine. So Notch was founded in 2018 out of the University of Toronto. And since then, we've been successful at raising a large amount of capital to grow the company over 3 sites. We're now over 80 people in Seattle, Vancouver, and Toronto. And the company now is really focused on end-to-end capabilities for producing therapeutics based on induced pluripotent stem cells. The company, the way I'll kind of describe the company is in 4 sections. One is that the company is named Notch because we are world experts in the control of Notch signaling during cell development. And I'll explain to you why that's important. We are, with regards to making T cells from pluripotent stem cells, we focus on creating function. So we want to optimize our uniformity and potency of the products we make in order to, have a vial full of T cells at the end that can be given to patients. Although this is an early technology, we are very focused on making sure we are focused on commercially compatible stem cell manufacturing. And I'll talk a lot about that, and how we are using induced pluripotent stem cells to maximize our opportunity for product designs that address the mechanism of action of the underlying pathologies that we're going after. Now, in order to understand why iPSCs can revolutionize patient outcomes for cancer therapeutics, we need to understand the limitations of current CAR T therapeutics. Now, those of you who are familiar with CAR T therapeutics, the process is quite complicated. You take blood out of a patient, you isolate their T cells, you use a virus to transduce those T cells with a receptor that targets the cancer, and then you give those cells back. It's about a 3-week process, and it's almost akin to like a self-organ transplantation. It's quite a complicated process. And what we can do with pluripotent stem cells is basically create an unlimited batch of cells that we then turn into T cells, which can then be filled and formulated and put into a vial to be given to a patient. So the first category is how we think about developmental biology. Adult T cells are limited by the composition and health of the donor. Whereas if you can make them synthetically from an induced pluripotent stem cell by understanding the developmental biology that's needed to take a cell from an undifferentiated state into a differentiated state, you can theoretically produce an unlimited supply of T cells. When you're deriving a cell from a donor, you don't really know what you're going to get at the end. And so you have unpredictable product composition. By creating them from a single source of pluripotent stem cells, we can enhance product efficacy and optimize the uniformity and potency of that product, so we know exactly what we're giving to patients. And manufacturing is a big one. As I described to you, the process for going from a blood-derived, a vein-to-vein model, they call it, is quite arduous. And in this situation, we're able to really improve the accessibility of the product by focusing in on the manufacturing. And then the last part is the limitations to what you can do to address the underlying mechanism of action. So we can deploy modern gene editing technologies to design pluripotent stem cells that are specifically enhanced to address the underlying mechanism of action of different liquid and solid tumors. So iPS-based T cells are relatively new in the industry. There's a lot of, there's a continuum from autologous, as I just described, to being able to take donor-derived materials that are and modify them so they're allogeneic, to the true pluripotent stem cell, which is kind of like the holy grail in that you can create unlimited batches and treat them like a drug. So this is there are no approved therapeutics based on pluripotent stem cell derived T cells yet. But the space is heating up and competition is heating up. And so it's a very exciting time to be part of this industry. Now, in order to make a T cell from a pluripotent stem cell, you have to do something quite complicated, which is that you have to recapitulate the development of a T cell, which normally happens inside the body in a dish. And what we know about T cell development is that when the T cell progenitors traffic from the bone marrow to the thymus, the developmental cascade absolutely requires Notch signaling. And the core of the technology that we licensed from the University of Toronto enables us to have full control over Notch signaling for producing these types of cells. The way people have been doing this in the industry for the last 20 years is using what we call plate-based approaches, which are really inefficient at introducing that Notch signal to developing cells. Notch signaling is not like a normal signaling pathway. Sometimes when we think about signaling pathways, we think about, throwing a ligand into cell culture, and that binds a receptor on the surface and it initiates a signaling cascade. Notch signaling is a little bit different. It's more of a mechanical force. When the two things come together, there's tugging and pulling and cleaving. And so you actually have to, you have to think about the biomechanical underlying of that interaction. So people have been using these feeder cells called OP9s that are very inefficient at inducing that Notch signal. And what we've done at Notch is we've taken a couple of pieces of technology and stitched them together to enable a Notch signal to be able to be delivered into a cell reliably and controllably. And the way we do this is by tethering the Notch ligands as well as an adhesion molecule onto the surface of beads, which enables us to have a tunable, precise, and predictable way of inducing Notch signaling in developing T cells in a manner that's scalable and commercially reasonable for manufacturing. So in this slide, which is covered by everyone's faces, is a little snippet of data to show the impact of the ability to control Notch signaling. And I'm focusing in on a particular part of the cell developmental pathway where we're going from a hematopoietic progenitor cell, which can give rise to many different types of blood cells, to a progenitor T cell, which is the cell that's ultimately going to give rise to a T cell. And what you can see is when we grow these on a 2D plate-bound system where we're using our engineered thymic niche technology to drive Notch signaling, we end up with a certain percentage of progenitor T cells. When we take that into a 3D format, when we put them onto beads and we can control we now have in our control the amount of beads and the concentration of those beads, we can drive up the cell type that we're interested in. But that's kind of not the big picture. The big picture is because this is developmental biology, when you're driving cells towards a cell type that you're interested in, you're also driving them away from the cell types that you're not interested in. And so if you look at the ratios of the cell types that we're interested in to the cell types that we're not interested in, we're enriching for the cell types that we're interested in. And so what this is enabling us to do is to be one of the few companies in the world that can actually coax cells towards the T cell phenotype in a dish. Now that matters because we can then take these cells forward and show that they actually behave like T cells in that they respond to T cell stimulatory signals. They express and secrete the things that make T cells kill. And we've shown in our labs, but we're not really presenting them publicly yet, that these cells behave the way you would predict they would behave. And so we're really excited about the future with these types of cells where we can now start thinking about the pipeline that we're going to build using this platform technology. As I mentioned, we're very focused on manufacturing. We've created clinical grade pluripotent materials. We've now been working at differentiating those cells towards T cells in sort of the clinical grade setting so that the rigor and reproducibility is there. So we're ready to take these products to the clinic when we're ready. And as I mentioned, we're developing a product pipeline. We have a partnership with Allogene Therapeutics, which is a publicly traded donor-derived allogeneic T cell company. We're working with them on using the pluripotent stem cell-derived T cells for treating hematological malignancies. Inside the company, we have our own internal program focused on solid tumor indications, where we're designing our cells to meet the underlying mechanism of action of solid tumors. I've said underlying mechanisms of action many times in this talk, but what does that mean? What does it mean when I say we're designing a sophisticated product profile? Well, what it means is that we're leveraging gene editing technologies to modify the genome of cells to achieve functionality in those cells. And some of these are listed here. So we can design exactly which binder we want to target the tumor. We can deliver payloads to the tumors to sort of modulate the tumor microenvironment. We can introduce safety switches that control the cells in the case of an unwanted side effect like CRS or tumorigenicity. We can prevent rejection by the host immune system, and we can prevent the T cells that we're introducing into the patient from causing GVHD, all by controlling the genome of pluripotent stem cells. We have an amazing leadership team at Notch. We have a lot of experience. Many of us have brought therapeutics companies all the way through trials. We have a tremendous amount of experience making pluripotent stem cell derived products. My previous company developed the first clinical grade gene edited universal donor cell. Chris, our SVP of research, was at Juno and Kite and led their preclinical program for pluripotent stem cell derived T cells. Emily Titus, Deepika Rajesh are extremely experienced in their field, and the co-founder Peter Zanstra helped build CCRM and is a professor at the University of British Columbia. So we have a really talented team. It's like playing in a really good band. As I mentioned, we're located in 3 centers of excellence. We have our headquarters in Vancouver, British Columbia, our manufacturing. We also have R&D activities in Vancouver. We have our manufacturing, and all of our clinical grade efforts are happening in Toronto. And we have our preclinical sciences team based in Seattle. And as a summary, we are really focused on maximizing the benefits of cell therapy by streamlining the regulatory, clinical, and commercial outcomes for cell therapy. We do this by creating unlimited amounts of T cells from pluripotent stem cells. We think this is going to revolutionize patient outcomes and increase accessibility of these types of therapeutics. We're looking forward to Bio this year. We're really going to seek out partnerships and new opportunities for our platform technology. If you have any questions about what we're doing, please reach out to me at gblock@notchtx.com. And looking forward to the meeting. Thank you again to the organizers for letting us speak at this event today.
Thanks very much, Greg. Fantastic presentation as always, and lots of exciting things going on at Notch. So please be sure to reach out to them when you're at Bio. The next presentation, I'd like to introduce Dr. James Halliwell, the Chief Executive Officer at Uraxia Pharmaceuticals. And James, the floor is yours.
Great. Thank you very much, Wendy, and to Life Sciences BC for hosting this great event. And it's such a pleasure to share the virtual stage with so many great local companies that we look forward to seeing around Bio and growing. So a little bit about Apraxia Pharmaceuticals. So first is the, there we are, move a slide here. So a few of the, we are a public company listed on the TSX, so a few cautionary statements. And then a little bit about us. So I was the co-founder of this company back in 2012. I'm a physician by background and actually worked at St. Paul's Hospital doing cardiac transplantation there as an anesthesiologist. And the critical thing when you're doing transplantation is it is all about precision. And drugs, as we all know, can be a little bit fickle. And so the company is really about taking the precision that we can achieve in an operating room and in a critical care environment and being able to get that stability of delivery, of getting the right dose of the drug to the right place for the right amount of time, but being able to do it really for outpatients in a variety of indications with high unmet medical need. So really, we are a technology company that is developing products. And so a little bit about the technology. So what you see on the top of your screen here is what I'll call sort of older technology. So when we look at a lot of depot technologies out there, they're often polymer-based. And so what they tend to do is take a whole lot of polymer and they mix it together with drug, and they rely on degradation of the polymer to release the drug that's trapped inside. What that does is when we think about almost every drug that we take, it's basically a mountain. So it goes from the bottom, hits a peak, comes back down. So it's this constant peak trough. What this old technology does is reduces the level of the peak and sort of widens the base of the mountain a little bit, but you still end up with this peak and trough. You also end up with a whole lot of polymer that your body then has to degrade and deal with the byproducts of. On the bottom, we really flipped conventional teaching upside down. So when you read the patents of almost all of the products that are sort of using the technology above, they will tell you, if you get below 50% polymer, drug release is immediate. When you look on the bottom, what you'll see is we use actually very little polymer. Our core is all drug. So we actually create a solid crystal drug core. We shape it, we size it the right way, and each of those are critical components in actually making sure that we're getting the right dose for the right length of time in the right place. The polymer is actually just a shell and does not degrade during the time that drug is present. That's a critical feature because we are actually using diffusion, and our body uses diffusion every single day incredibly efficiently and effectively. And we are harnessing diffusion in this instance in order to be able to deliver drugs with great precision, great locality, and to be able to also get the duration that we want. So when we look at this, we say, how can we apply this technology and where are the products that we can use this for? So one of the first places that we looked is actually knee osteoarthritis. So why did we look there? So knee osteoarthritis has had so many attempts at very sort of fancy ways to do it, whether to grow cartilage, whether to use biologics to try and treat the pain of osteoarthritis and slow the progression of the disease. And really, most of them have met with failure. The one drug that has remained persistent for 50 years are corticosteroids. They have been injected into the joint and provide very effective pain relief. However, they don't last very long. They do cause cartilage damage, meaning that they're really reserved for infrequent use at the end of the disease. Prior to joint replacement, and they also cause your body's natural steroid cortisol to be suppressed. Despite all of these different therapeutic options that have been brought all the way through phase 3 and failed at that point, the American College of Rheumatology and the American Academy of Orthopedic Surgeons only recommend 2 pharmacologic sort of agents. One, intra-articular steroids, which is what we are at our core. And then in addition to that, oral non-steroidal anti-inflammatories, which are really for mild disease. So our lead asset, EP104-IAR, we have taken a steroid, fluticasone propionate, which you may know as Advair, Flonase, and we're the first to ever turn it into an injectable. Because of that, we're talking about a generic product, but we have composition of matter claims that are in our granted patents all throughout the world because of what we actually do in turning it into an injectable and putting it in our technology platform. So one of the key things when you look at the success of a product in osteoarthritis is that number one, it needs to treat pain, but safety is also incredibly important. And remember, osteoarthritis is a very complicated disease that is damaging cartilage. So you do not want to give a drug that actually damages cartilage. Intra-articular steroids, across all of them that have been given that are approved, they damage cartilage. So when you give them in the study that the FDA requires, the demonstration of safety, they use something called the Mencken score. So this is actually done in dogs, and dogs unfortunately have to be sacrificed in order to be able to look at the effect on the cartilage. And basically any score greater than 0 means you're damaging cartilage. The standard scores will go to 2, 3 as means with much higher scores. And the whole reason for that is the burst. So it's the peak of that mountaintop that the steroids deliver. And it's a class effect. If you take our drug and you deliver it in a standard technology, you will cause cartilage damage. So this is not about the drug. This is about our technology. What you're looking at here on this slide is a very large study in 216 dogs that we did for a year and then took this to the FDA. The middle dose of 5.5 milligrams equates to what we are using right now in our first pivotal trial, where we expect data in Q1 of 2023. You can see that at every dose, including the equivalent of giving 5 years of dosing all at once, we preserve cartilage. So we have 0 cartilage damage. This is incredibly important. And very interestingly, as the years have progressed, there's been an incredible recognition at just how important cartilage damage or lack thereof is in the treatment of osteoarthritis. And we really hold a very unique position here with this kind of data. After that data, we went and we did a phase 1 study. This was a small trial of only 32 patients, 24 active, 8 placebo. We followed them for 42 weeks. It was incredibly well tolerated systemically, locally. We saw that we had controlled, targeted, and very predictable release. We also saw immediate and sustained pain relief. And based on that, we then went into our phase 2 trials. So we have both FDA and European approval. We are running this trial actively at 12 sites right now. And you can see it's a very large sort of phase 3 in size, 150 patient per arm trial. And we are following patients out to 24 weeks, looking to provide 6 months of effective and safe pain relief. So as we look to see that data in Q1 of 2023, which obviously will be a major inflection point for us as a company and in that asset, the other thing to just highlight is, again, we are a technology company here, and we have the ability, and we will be talking very soon about other products that will be coming into the pipeline, not just osteoarthritis, but also in gastroenterology, infectious disease, and in oncology. Imagine being able to deliver the therapeutics that you want, that you know target the tumor cells directly into a tumor for a long period of time, very precisely, without systemic absorption, so you can target that tumor without having all the damage of that drug to the rest of the healthy cells in the body. With that, I just want to highlight our board of directors. You will see it's obviously a very VC heavy board of directors, but these are some of the people who've really delivered the biggest successes in VC. So Simon Pimstone, who's the founder, executive chair of Xenon Pharmaceuticals, Richard Glickman, who's been the founder and chairman of Aurinia, of Espriva, and is a venture partner at Lumira Ventures. Again, incredible success in biotech from both of those individuals. John Montalbano, who used to run RBC Global Asset Management and sits on Accelera's board, Uraxia, and the Canada Pension Plan Investment Board. And Paul Geyer, who brings an incredible amount of success from VC-based companies, particularly in the medical device space. So with that, cognizant of time, I just want to thank Life Sciences BC. We are happy to address any questions, look forward to meeting with people and really developing partnerships as we grow. So thank you very much, Wendy, and to all of you for attending.
Thanks very much, James. Great presentation, as always. I would like to now welcome Handel Kim, co-founder and CEO of Variational AI. Variational AI is building a discovery engine that will rapidly generate novel and optimized molecules for unmet medical needs across disease areas to redefine the economics of drug development. Over to you, Handel.
Thank you very much, Wendy. Okay, can people see the screen? Okay, thank you. My name is Handel Kim. I'm co-founder and CEO of Variational AI, and it's a great pleasure to be able to speak to everyone today. So first of all, about Variational AI, we're based in Vancouver, founded in September 2019. The leadership team has proven track records in both machine learning and drug discovery. We have top machine learning expertise with team members from institutes such as MIT, Caltech, Sharington University, and UBC, as well as previous work experience at Microsoft Research, Google Research, and D-Wave Systems. We also have extensive drug discovery success, 124 patents, 11 drugs taken into late-stage clinical trials, and multiple drugs licensed to pharma. We are an AI for drug discovery company, and we validated our approach, our algorithm and platform in partnerships with various organizations. We've succeeded in attracting seed investment in the total of $3.5 million US from leading investors in Seattle, Silicon Valley, as well as here in our backyard, the UBC Seed Fund. And we also were able to obtain non-dilutive funding from the Digital Technology Supercluster. So our big, hairy, audacious goal is that we are building an AI-based engine that rapidly generates novel and optimized small molecules for unmet medical needs across disease areas. And our goal is to redefine the economics of drug development. It's a big goal, and it's something that we've made progress on, but it is our grand mission. You have to start somewhere, and our discovery engine, Enki, is what we call it. We're starting with kinases. So let me take a step back, because we are an AI for drug discovery company, and there may be people who don't fully understand AI. Sometimes AI doesn't understand AI, but really we pose the problem very simply as this. We're in the business of discovering molecules. We solve problems in chemistry. And for the chemists in the room, there's a concept called chemical space that you'll be familiar with. And chemical space is essentially a universe of 10 to the 60 drug-like molecules. So that estimate varies, but it is literally a universe. In fact, it is a billion times, a billion times, a billion times, a billion times bigger than all of the stars in the visible universe. The fact is that science has only explored a very, very small fraction, maybe a few 100 million on a good day, maybe up to 1 billion molecules, where we actually understand the relationship between the structure of a molecule and the properties. That is effectively 0% of chemical space. And so we're driven by the motivation of trying to understand the question, how many life-saving blockbuster drugs lie in chemical space just waiting to be discovered? Of course, with such a massive search space, it's impossible to search through brute force. The main problem here is that it's not just a vast search space, but it's extremely chaotic in the sense that the structure of the molecule, which informs its activity, and the property are not necessarily correlated in a good way. So you can start with a molecule with well-known properties and then search around it and find that the other molecules that are extremely similar from a structure basis are radically different when it comes to drug properties such as potency. Very simply put, it's like searching for an address using a phone book. So chemical space can be arranged based on structure. Similarly, you could have a phone book with 10 to the 60 entries all arranged in alphabetical order, but that doesn't help you find where M. Smith is located. Rather, what you want is a map. What we have done is used a generative machine learning in order to transform the space of structure into property. When we use Enki, it's like searching for an address on a map, where spatially based exploration is intuitive and efficient. More specifically, if you're looking for an address, using a map makes sense. If you're trying to find a drug, you should try and use properties rather than structure. So status quo chemistry use the approaches search for drugs with specific properties based on molecular structure. On the left here, you'll see a series of molecules, and they look extremely similar. And this would represent a very similar, a very structured and very incremental difference and change across the spectrum of these molecules. Yet at the same time, the values associated, in this case, these are docking scores, change fairly dramatically. So this molecule and this molecule are almost structurally identical, however, may have very different properties. What Enki does is, as I mentioned, we transform the space from structure into properties. As a result, when we rearrange chemical space, we're able to put molecules that are dissimilar structurally, but uniform with respect to the properties, and that's what you want. The fundamental question is, if drug properties are what you're looking for, then why not search directly in that space? And that's effectively what we do. And what we're doing is we're going after kinases as the initial target class that we're training our discovery engine on. And why kinases? Well, there's well understood biology. We work in chemistry. We're looking for targets that have relatively de-risked biology. We look at kinases because there are 71 FDA-approved inhibitors for various targets, 110 in clinical development. What this means is that there's ample training data that we can avail ourselves of. And that's very important because at the end of the day, we are an AI company. The other thing that you find is when you look at the approved drugs, as well as a lot of the drugs in development, the scaffolds of these drugs or of these molecules are not diverse. They cluster generally. And that's not necessarily surprising because kinases themselves, these protein kinases, are structurally quite similar. One of the main challenges in developing small molecule therapeutics for kinases is selectivity. You might have this ligand that binds to one kinase, but it'll bind all over the place. And controlling that selectivity is extremely important. And that is exactly what our machine learning model is able to, and we validated it. So being able to control selectivity of a small molecule to an insane degree allows us to generate molecules that are extremely selective and have the cell penetration to be able to go into solid tumors, as opposed to biologics which have the selectivity and specificity, but may not be able to have the same cell penetration. Fundamentally, the similarity of the multiple kinases, and there are 516, I believe, in the human kinome, the similarity and the problems that make it very difficult for traditional drug discovery efforts makes it actually very good for us using our machine learning approach. Very fundamentally, very simply, what we do is we train our algorithm on data sets. They're public data sets, commercial data sets that we've obtained and generated in silico and computationally. We train multiple kinases, and there's a lot of data for each kinase. And because of the similarity, we're able to train multiple kinases to create a unified model for kinases that can then rapidly generate high quality leads for a large range of kinase targets. And as I mentioned, we're going after oncology. Once we've built the model, which is currently in development, we should be able to generate optimized leads optimized for multiple properties. And we do this simultaneously: potency, synthesizability, ADME, tox, et cetera, in about 2 weeks per target. This is potentially very transformative in terms of we don't need to retrain the model, but we can go after multiple kinase targets with the same trained model and generate massive value. Our goal at Bio essentially is to do business development. We're seeking collaborations and partnerships with biopharmas working on multi-targeted kinase inhibitors. And we look forward to joining the BC delegation. Thank you very much for your time.
Thank you very much, Handel. As always, a fantastic presentation and lots of exciting things going on with your organization. I would now like to welcome Karim Walji, CEO of Microbion Pharma Corp. Microbion is a clinical stage biotechnology company dedicated to the development of its proprietary antimicrobial antibiofilm family of compounds as therapeutic agents for the treatment of infectious diseases, including those caused by antibiotic-resistant bacteria. Over to you.
Great, thank you so much. It's a pleasure to be here. I'd like to thank Life Sciences BC for the invitation. I think it's a great program. And we are very much looking forward to our meeting at Bio. We're looking primarily to raise funds and basically create partnerships as well. So those are our two objectives, and that's why we are going to be there. With regard to, sorry, I'm having trouble advancing my slides. And I will ask Claire to take over, but I will just start over by framing the company. We at Microbion are developing a new class of anti-infectives. It's basically a class called the Bismane. The WHO has given us a new stem, which basically recognizes us as a new class. And we basically have 3 active programs. We have one phase 1 program, which is a program in treating rare orphan diseases. So we have just finished our GLP talks and are now going to be entering a phase 1 study to advance our program in something called NTM, non-tuberculous mycobacterium infections, and CF infections. We also have a two phase 2 programs, and I'll show you some data from one of those programs in treating diabetic foot ulcer infections, as well as orthopedic infections, device-related infections. And Claire, if I could ask you to go forward 2 slides, please. The one thing that's important about Pravibismane, which is the name we were granted by WHO, is that we not only are a new class, but we have a novel mechanism of action. And the important feature of that mechanism is the fact that we not only are able to have a broad spectrum of activity to eradicate all the superbugs or the priority pathogens identified by the CDC, but we're also able to eradicate the biofilms. And the biofilms we feel are very important to managing multi-drug resistant infections or difficult to treat infections. infections, and I'll speak to that in a few minutes as to why that's so important. We have run 5 clinical trials today. We have experience in over 328 human subjects. We have also seen signals of efficacy in the 2 clinical studies that we've done in both diabetic foot ulcer infections and orthopedic device-related infections. We have received a lot of support from key stakeholders, non-dilutive grants from the Cystic Fibrosis Foundation, from CARB-X, from NIH, over $21 million to date. And we are developing an anti-infective, and that's really not necessarily an area that's in vogue nowadays. It's been tough, challenging for some companies in this space, which is why very early on we decided to focus on a local indication strategy. So we are focused on not systemic applications, which we believe have multiple benefits, which I'll also get into in a few minutes. And we also have strong IP protection, with our most recent IP being granted that takes us to 2041. And in addition, we have orphan designation. We have something called QIDP designation that can give us up to 12 years of protection independent of IP. Next slide, please. I have mentioned that we are doing 3 indications. I'll just move to the next slide to provide some more reasons and logic for why we are taking a local administration. We have a local for treating DFI. It's a topical drug to treat diabetic foot ulcer infections. We have a local administration at the surgical site to treat orthopedic infections. And we have an inhaled formulation for treating lung infections topically to the lung. And there are multiple advantages to that. By actually having focusing on the indications that we are, for example, DFI, cystic fibrosis, non-tubercular mycobacterium, these are actually not hospital-based indications. So number one, we don't have the hospital control issue, but we also are able to get the drug to be used in a very targeted way that also prevents off-label use, so to speak, and limited usage outside of the indication. Also, by treating locally, you have a reduced risk of resistance because you're only getting the drug to the local site without systemic absorption. You have less risk of exposure that creates the environment for resistance. We also have, we believe, an improved safety profile, again, because you're not going systemically, and better efficacy because you're going to the site of the infection itself. I'll move to the next slide, please. I just want to quickly touch on biofilms. As I mentioned, we're able to eradicate not only the bacteria, but also the biofilms that are formed by the bacteria at equipotent doses. And that's very important. Very often, if you can even get rid of the biofilm, you'll need much, much higher doses. We can do it at equipotent doses. And biofilms protect the bacteria against host immunity and also from the effect of antibiotics. And they're involved in 80% or more of human infections. And what's also very important is biofilms are, an environment that create an environment for resistance. So there's up to 1,000-fold increased rate of mutation in biofilms. And it's also important to note that the most multi-drug resistant pathogens are also the most prolific biofilm formers. So our new class with our new mechanism that allows us to eradicate both the bacteria and the biofilms, we believe is very important for unlocking the potential to treat some of these serious conditions. Next slide, please. The first study we did was in diabetic foot infections, obviously a big problem, and I'll go right to the next slide in the interest of time. But I wanted to quickly show you the results of a study we did. This was a safety study where we looked at efficacy signals in patients with diabetic foot ulcer infections. These patients were treated topically 3 times a week for only 3 weeks. And in the first picture, as you can see on the left, that is what the wound looked like at the beginning of before the patients were treated. The second picture is 2 weeks after last treatment, or roughly 5 weeks. And the third picture is 4 weeks after last treatment, or 7 weeks after initial treatment. And you can see a significant shrinking of the wound. So basically, we have seen an 85% reduction in wound size versus 30% for placebo. Everyone was getting standard of care, by the way. And we also saw a 6-fold reduction in amputation rates. The ends are very small for the amputation rate, so obviously we need to do more work, but directionally we saw some very interesting results. If I could move to the next slide, please. We also are treating inhaled infections. There's a company called Insmed that developed a drug called Arikayce. They have generated a $3 billion market cap, and we have data that basically indicates potential advantages on many fronts, including efficacy, including safety. That company has a black box warning for their product, and we have very low resistance potential as well, and also a very convenient dosing regimen of perhaps once daily or perhaps even once weekly. Next slide, please. I won't go through this in detail, but we have we've just finished our GLP tox studies. We've done some very, very interesting in vivo studies for our last formulation, which is the inhaled. And we're starting human trials on that indication in January. And what I can say is for all the indications we are pursuing, our commercial potential is quite significant, roughly $1 billion for each one of those indications. Next slide, please. And as I mentioned at the beginning, we're preparing for a Series B raise. So we're looking to raise money to advance our programs, especially the diabetic foot ulcer infection program and the inhaled program for treating the lung infections, the orphan lung infections. And we're also seeking global and regional partnerships as well as a way forward. So I'd like to thank Life Sciences BC and all of you for your time. And in the interest of time, I will skip the last slide.
Thanks very much. Great presentation, as always. Look forward to seeing you next week at Bio. I would now like to introduce Yvonne Martins, Executive Director, Providence Health Innovation, Research and Engagement, Providence Research. She's going to be speaking about the developments within her organization. Providence Research is the research partner of Providence Healthcare, one of Canada's largest faith-based healthcare organizations, operating 17 healthcare facilities in Greater Vancouver, including St. Paul's Hospital, which enjoys a reputation as a world-class place to conduct biomedical research. Yvonne?
Thank you. Good morning. I'd like to start by acknowledging with gratitude that I'm joining today from the traditional, ancestral, and unceded territories of the Musqueam, Squamish, and Tsleil-Waututh nations. It's also the home of the new St. Paul's, currently under development here in Vancouver, which I have the privilege of introducing to you today, with the goal of developing collaborations, partnerships, investment, and other contributions so that we can truly optimize the potential of this unique opportunity to deliver a leading edge platform for collaboration between public and private sectors. So the new St. Paul's will be operated by Providence Healthcare, and it is a 17-site health system serving the entire province of British Columbia and beyond. And with the new St. Paul's, we're working to build on our track record of having global impact. So some examples of this kind of activity that we've had are here. On the top left, you can see work at St. Paul's has been instrumental in taking HIV from a death sentence to a manageable disease. And on the top right is Dr. Don Sen with our Center for Heart-Lung Innovation. He's the most highly cited researcher in the field of chronic obstructive pulmonary disease in North America and second in the world. He's recently done important work with COVID, from developing a new COVID test with academic partners and working with Vancouver Airport and an airline and implementing rapid testing, and to helping lead in the development of a provincial long-term care clinic for COVID patients. On the bottom right, you'll see Dr. John Webb and his team. They're pioneers of transcatheter aortic valve implantation, who, working with industry in valve development and testing, have made open heart surgery no longer necessary for valve implantation. So it's really this kind of impact that the new St. Paul's is going to amplify and accelerate. And here is the location of the current St. Paul's. It's indicated by the icon on the top left in beautiful Vancouver's downtown. And the new St. Paul's is less than 5 kilometers away. On the bottom right, you can see the icon there. Here you can see the new St. Paul's is the health anchor for a 450-acre City of Vancouver plan for synergistically focused businesses in the life sciences sector. BC's Innovation Commissioner has said that potential exists to develop a major globally recognized health R&D flagship precinct as part of the development of the new St. Paul's Hospital and Health Campus. The area already has a strong academic presence, and with the new St. Paul's and AbCellera's current development project, we have the largest public and private biotech life science medical projects currently being developed in Canada, located within a mile of each other. So the St. Paul's Hospital and Health Campus will be a world-class integrated healthcare research, teaching, and innovation hub. We have the rare opportunity to build from the ground up. It's a $4.2 billion project at this point. Here you can see our vision for the 18.4 acre site. The acute care hospital at the top of the screen will open in 2027. It's well underway, almost at ground level in terms of construction. The Health Innovation Hub on the right-hand side is our focus now, and it will also be open in 2027. The rest of the site will be planned out after that, but you will see on the left-hand side, there will be additional development for commercial industry partners. Here we have a proportionate representation of the hospital and Health Innovation Hub. The big red circle is the acute care hospital and ambulatory care. It's massive, around 1.4 million square feet. And importantly, it connects by Skybridge to the Health Innovation Hub. And in the Health Innovation Hub on the right-hand side, you can see the gray dot. That's the largest amount of space. It's reserved for industry partners. We'll just take a little bit of a deeper dive into the purple dot, the research labs. This is how we're anticipating the research facility to be proportioned. 180,000 square feet of the 840,000 square foot Health Innovation Hub. That's roughly 50-50 for dry lab and wet lab research, helping fill the really huge demand for wet lab space in the region. You can see that we have everything from preclinical through all the wet labs, so histology, biobanking, the dry labs that include space for independent clinical researchers, which are practicing physicians in renal, emergency, orthopedics, and other departments that industry will have the opportunity to partner with. And also, for example, the shared data management facilities, capacities, that really will be the drivers for research, innovation, and entrepreneurship, and they'll be embedded throughout the building and the hospital. So this is going to make it possible for us to build on our track record of being able to solve complex local problems that have global impact. So for example, to really change HIV, we needed everything to translate research discoveries into frontline clinical practice, from the patients, biobanks, data, wet labs, the ability to genotype right through epidemiology and statistics, and the ability to really run health economics to inform policy. And that's why the United Nations model for responding to HIV worldwide came out of St. Paul's, because we had everything together and we successfully worked with partners, which included government, industry, health authorities, healthcare providers, academics, and communities. We're now working to use that same approach, for example, in tackling addiction, because we're absolutely at ground zero for that. And we have leadership in that arena that we're going to expand on. We think we can do the same in our other research and innovation focus areas. These are areas that most affect the communities we serve. They're the areas that are the world's greatest health burdens, challenges. This also means these are areas that hold great opportunity for industry in working with us to develop solutions to address those challenges. These are areas where we have real research and innovation strength, reinforcing clinical activity that's very intense. We are, for example, the Provincial Center for Cardiac and also the Provincial Center for Renal. We have real leadership also in the area of elder care. We have the Healthy Aging Center and the first publicly funded dementia villages in Canada. Providence Healthcare includes hospitals as well as long-term care facilities. And here you can see some of our emerging areas of clinical and research expertise, where we're making a really concerted push in developing, so they'll be up and running by the time we're in the Health Innovation Hub. VR simulation, as an example, we're working on the business plan now with the aim to roll out VR into homes in the community. In our dementia villages, for example, this represents phenomenal opportunity to study patients in a real life environment, to understand the impact of digital technologies, for example, which will be important to industry, and really to provide patients with a precision medicine approach, which is where we're aiming to be. So now we'll take a closer look at the Health Innovation Hub itself. With the facility and capacities here, combined with a very focused effort on partnership, A recent demonstration of our commitment to this is Providence Healthcare. While we're not industry or private, we're actually one of the founding members of Canada's Digital Technology Supercluster. With the Health Innovation Hub, we're creating a platform to co-develop solutions with partners. Engagements well underway with industry, academic, and other partners, even including input into functional planning. The facilities you see listed here are things that both the public sector and the private sector need. What's so enabling and unique is, crucially, we have patients. There's the SkyBridge connecting the acute care hospital with the Health Innovation Hub, with the hospital and medical offices on the right, to our research, innovation, and industry partner space on the left. The SkyBridge is part of a very intentional strategy to break down silos and facilitate interactions between physicians, researchers, students, staff, patients, industry, and other partners. It will create a real-time link, bringing research from the lab back to the patient faster than ever before. And here you see there's things you would expect: a conference center, corporate services, but at the heart of it is an innovation center around 25 to 30,000 square feet. And it will consist of sandbox facilities, prototyping facilities, ideation space, formal and informal meeting and collaboration space, because we know that discovery and innovation grows in the gaps between the disciplines and between organizations, and that's where the fertile spaces are. And that's what the Innovation Center is designed to create, that true collaboration and partnership. There'll also be a whole suite of core facilities around the work we do that will be accessible to our partners. We will have the first in human clinical trial center west of Toronto that's not cancer specific in the hospital. It will be linked operationally to the Health Innovation Hub with small scale biomanufacturing and also expanded clinical trial activity. Currently, we have early phase 2 and phase 3. This year alone, we actually have 1,100 clinical trials underway. Other facilities will include nanotechnology, regenerative medicine, 3D bioprinting. So we just take a closer look at one of these facilities or these capacities. We are developing a data access and services center, the physical and digital infrastructure needed to move evidence into practice in real time. The center will collect, process, and analyze patient data even from beyond the hospital walls, for example, at home, in the dementia villages, long-term care facilities. And even more differentiating, we'll be building on our strength with our biobanks and linking biospecimens to data. We've already developed the Integrated Health Informatics Data Lab, and our first customer is Genome BC. And using this, they've just launched $1 million health data program for researchers. So the data lab will really allow researchers and innovators from private and public sectors around the globe to access the real world evidence. They need to support healthcare discoveries, product development, and ultimately better care for patients. So just to knit together how all these parts fit in the Health Innovation Hub and with our partners, for instance, we see what the problem is because we have the patients who are suffering, and then using our data, our biobanks, and in partnership with academics, industry, and public sector, we can work on a solution. We can prototype it in our Makers Lab. We can trial it through our phase 1, phase 2, and phase 3 clinical trials. We can incubate it, invest in it, commercialize it. And companies large and small can be on site and have access to the leading edge core lab facilities and without having to purchase them for their own. And also while sharing these facilities, we know that collisions will happen and partnerships will be built. So I'd like to close by inviting those interested in working together now and going forward onto the site to be in touch. Together, we can really massively leverage this investment and the opportunity and outcomes with the new St. Paul's. And thank you for this opportunity.
Thanks very much, Yvonne. Super exciting. And we look forward to continuing to work with you on this initiative as it continues to move forward. Our next speaker is Julie Rezler, Vice President, Corporate Development, Primary Peptides. She's going to be speaking about the latest work from their organization. Primary Peptides is a private clinical stage protein manipulation company focusing on hard-to-drug targets. Thanks, Julie.
Thank you, Wendy. Okay. Thank you very much, Life Sciences BC, for the opportunity to present today. And also I'd like to express my sincere gratitude for the leadership that you are showing in supporting the BC Life Sciences companies at BIO this year. It's a privilege to be able to introduce you to Primary Peptides today. Here's my agenda, where we only have a few minutes together today, but I'd like to give you a high-level overview of our company, the problem we are addressing and our solution using our platform technology in protein manipulation. I'll provide a highlight of our pipeline, our current partnerships and assets available for licensing, and I'll sketch out our plan for our Series A financing, and I'll close with a slide on our accomplished team. I think it's important to highlight our objectives for Bio this year. We are keen to meet with companies for collaborations for our assets and our platform, as well as meeting with investors for our Series A, $25 million for a 3-year runway. So Primary Peptides, we are a spin-out of the Faculty of Medicine of UBC. We are a clinical stage protein manipulation company focused on developing first-in-class assets targeting diseases of unmet medical need. We have a rich pipeline consisting of 7 assets in total, 3 in cardiovascular, 2 in oncology, and 2 in neuro. And as you can see from the slide, we currently have a clinical asset that's completed a phase 1A and is advancing into phase 1B/2A in acute stroke patients. And we have 2 assets in IND-enabling studies in acute stroke and acute myocardial infarction. Our preclinical assets are all available for licensing. We have signed 3 drug development partnerships with Greater China for Greater China rights, but rest of world rights are available. And we have a strong patent portfolio and a clean cap table. So what is the problem that we are addressing? Only 15% of the human proteome has been drugged to date. 85% of the human proteome remains undrugged. And as you know, most drug targets are proteins, but most proteins cannot be drugged using conventional methods. And that's why we are focusing on peptides, as they sit in the sweet spot between small molecules and antibodies. We are expanding the pool of druggable targets using our proprietary platforms, and we do this in 3 ways. We're able to block the interactions of proteins. We're able to stabilize the interactions of proteins using our molecular glues, and we degrade proteins. Currently, the protein degradation space is dominated by PROTAC companies, and so we are a newcomer into the protein degradation space, but we have carved out a very solid niche. Our technology is differentiated from PROTAC technology in 3 important ways. Our peptides are bifunctional, meaning we can combine a protein-protein interference blocker with a protein degrader in the same peptide therapeutic, achieving dual action. So what that means is that we block a disease-causing pathway and we degrade a disease-causing protein using the same therapeutic peptide. Also, we can degrade proteins via the lysosome or the proteasome. And these are the natural degradation machineries of the cell. And we conduct experiments to determine which is the better disposal mechanism to use. Plus, we can apply our technology to gene therapies, whether it's messenger RNA, LNP, or viral vectors. And compared to other modalities, our therapeutic peptides are highly selective. They are very low, they have low toxicity because they're composed of natural amino acids. And our peptides are both cell permeable and able to cross the blood-brain barrier. I appreciate this is a rather busy slide, but it does show our 3 interrelated platforms. It's important to point out that our key foundational advantage is our ability to precisely identify the exact contact points of any pair of proteins. If you look on the slide, if you look at the diagram on the bottom left, this is our protein-protein interference technology. This green neon stick figure represents our peptide therapeutic. It is rationally designed to fit neatly into the exact contact points of a protein, blocking its interaction with its green protein to its left. A small molecule would be a tiny dot on this protein, and an antibody would be exactly the same size, if not larger than the protein. Our molecular glue technology, the diagram number 2 in the center, using our molecular glue technology, we are able to induce proximity between 2 neighboring proteins which results in stabilizing and strengthening the interaction between these two proteins. Our third technology is what we call ChapTag, which is our degradation technology. This is our therapeutic peptide that is outside the cell cytoplasm, and it consists of three domains. The cell penetrating domain, which crosses the cell cytoplasm and is able to cross into the blood-brain barrier. We have the protein binding domain, which seeks out a target disease-causing protein and forms a peptide protein complex. The third domain is our lysosome-proteasomal degradation targeting motif, which traffics the disease-causing protein and peptide complex to either the lysosome or the proteasome for degradation. Our peptides are rationally designed. We do not need to know the protein structure. They have normal stoichiometry. There is no catalytic process or E3 ligases that is typical of PROTAC technology. Our pipeline has 7 first-in-class assets, all focused on hard-to-drug targets, and they've been developed using our protein-protein interference blockade technology, as well as ChapTag, our degradation technology. As you can see on the slide, some are interference peptides and others are degrader peptides. These degrader peptides here for acute stroke, resistant prostate cancer, liver cancer, and Parkinson's disease are bifunctional, meaning that in a single therapeutic peptide, we can block the disease-causing pathway, as I mentioned earlier, using interference peptides, but we combine that with a degradation peptide to degrade the disease-causing protein. Our 3 CVD assets, our cardiovascular assets, have been currently partnered with Chinese companies, all very well known and large. The top 2 are publicly traded, Prime Medicine is private. Rest of world rights are available for partnering. All studies are being conducted to meet the standards of the Chinese NMPA and the US FDA. So what this means is that, for example, K13, which has completed a phase 1A study with positive data, is planned to move into a phase 1B/2A study in acute patients later this year. If that data is positive, we have been assured that we can file an IND and progress into a phase 2 clinical study without necessarily conducting additional clinical studies. Our remaining 4 assets, our preclinical assets, are all available for licensing. We have statistically significant in vivo data for all these 4 assets. Our liver cancer asset and Parkinson's disease asset are ready for IND enabling studies. For resistant prostate cancer, we have a tumor regression study planned for later this year. And assuming that data is positive, it will be ready to advance into IND enabling studies. And TDP-43, we are conducting additional in vivo efficacy studies. And assuming they are positive, the asset will move into IND enabling studies. We are seeking a Series A of $25 million for a 3-year runway. This will be the company's first raise of dilutive financing. So far, we have been supported by partnering revenues and grants, and we have a very clean cap table. This infusion of cash will enable us to take some of our assets to key inflection points to facilitate entry into the clinic as well as for partnering. The 4 preclinical assets that I mentioned earlier, we will prioritize the ones which we would like to advance into IND enabling studies, and we will support 1 phase 1 clinical study with this raise. The other 2 preclinical assets, assuming we're successful with positive data that move into IND studies, we will seek partnerships for them. The CVD asset that I mentioned previously, if that is able to go into phase 2 directly, we would like to partner the phase 2 clinical study for rest of world rights. The funds will also go towards supporting and enhancing our platform. We are investigating different formulation options to improve the administration of our peptides, which are currently administered once a day using IP or sub-Q. And we would like to extend the administration to once a week once a month, or even oral. Plus, we would like to also spend funds to refine and further develop our molecular glue technology, as well as enhance our nucleic acid delivery. We are a small and nimble team, and so the funds will also go towards expanding the team in order to be able to drive more value within the organization. And speaking of our team, we expanded our management team recently this year, and we also expanded our advisory board, appointing experts in drug delivery, drug discovery, drug development, cardiovascular trials, as well as Alzheimer's disease. So I'd like to thank you very much for the opportunity to present today. It's been a pleasure. Here are my contact details below, and I'd be happy to take any questions in the Q&A function. Thank you very much.
Thank you very much, Julie. Great presentation. And our next speaker is going to be Dr. Robin Sharington, Vice President, Strategy and Innovation from Xenon Pharmaceuticals. Xenon is a clinical stage biopharmaceutical company committed to developing innovative therapeutics to improve the lives of patients with neurological disorders. Over to you.
Okay, thank you. Thank you so much to the organizers for inviting us to present during the BC showcase. And obviously it's my pleasure today to do a brief introduction to Xenon. But before I get into the details, I do need to make everyone aware that Xenon is a publicly traded company under the Xene symbol. And this presentation will have certain forward-looking statements that come with certain risks, uncertainties, and assumptions. So Xenon's vision is to become a company that better treats neurological disorders. And our strategy to attain this is to become a fully integrated pharmaceutical company with offices both here in Vancouver as well as in Boston in Massachusetts. And currently we're around 200 employees. What I would like to do is just touch a little bit on our pipeline. Our pipeline, as you can see from this slide, has a mixture of proprietary products as well as those that have been formed partnerships, and those partners are now responsible for the development of those products. Our proprietary products are very much focused on ion channel modulation of the Kv7 potassium channel. Kv7 potassium channels play a critical role in the control of excitability in the brain. And by opening these channels with small molecule modulators, you're able to control the hyperexcitability that underpins certain clinical disorders. Xen 496 is a pediatric friendly Kv7 opener that's currently in phase 3 development, a single phase 3 study for a rare genetic epileptic encephalopathy, a devastating epilepsy that presents in the first few days of life. And it's in its phase 3 clinical study of approximately 40 subjects, and it's called the EPIC trial, and that is underway. The genetic underpinning of KCNQ2-DE is caused by loss of function mutations in the gene, and the gene is translated into the Kv7.2 protein and depletes the potassium current. 496 as an opener of that channel is able to then replenish the missing current, and therefore it's acting as a precision medicine targeting the genetic root cause of that devastating epilepsy in young children. 1101 is a next generation potassium channel opener that we have currently that's concluded a large phase 2b study in adult focal onset epilepsy, a prevalent epilepsy condition. And we're also exploring its utility as a treatment for major depressive disorder, including major depressive disorder with anhedonia. Anhedonia is a lack of ability to feel pleasure. And we have 2 studies underway. One is with Mount Sinai, looking at the functional activity of 1101 in the brain reward system in MDD patients. And we're looking to initiate a clinical study ourselves, looking at clinical endpoints of depression as well as anhedonia in major depressive disorder. The mechanistic rationale for 1101 in MDD is based both on animal data showing a depletion of Kv7 activity in the reward system in certain animal models of depression and anhedonia, as well as clinical studies from a first generation Kv7 modulator that was shown to reverse the depression in MDD patients and improve their hedonic capacity. I'm going to touch on a little bit on the XTOL data, the phase 2b data for 1101. This was a randomized, placebo-controlled, double-blind trial where 1101 was dosed at 25 mg to 10 mg, and there was approximately 320 subjects were enrolled, with 100 in the 25 mg dose group and 100 in the placebo group, and roughly 50 in the other two groups. Patients were dosed for 8 weeks and then went into an open label extension where we're beginning to collect long-term, greater than 1 year safety data for 1101. Just to touch a little bit on the demographics and baseline characteristics, this study was actually conducted obviously during the COVID pandemic. And if I can draw your attention to the bottom line, these patients which enrolled in our study were significantly refractory. You can look at the number of previous anti-seizure medicines that these patients have failed, and you can see the medians are between 6 and 5. And when you consider that after a patient has tried and failed to get their seizures under control with 4 ASMs, it's extraordinarily difficult to get the patient then to have seizure control. You can see that we enrolled a very refractory population into our clinical study, and it's significantly more refractory than the clinical studies for pivotal studies of currently approved ASMs. Despite this, we saw robust efficacy, both statistically significant for all of the doses, 10 mg, 20 mg, and 25 mg, and the result of 52.8% median percent change in 28-day seizure frequency is significantly greater than is typically observed with the currently available ASMs. Their pivotal studies typically had MPC changes of between 30 and 40%. So we see this as evidence that the Kv7 mechanism is able to generate further improvement of seizure control, particularly in this highly refractory population. What was also nice to see that there was consistent data for 1101 looking at the responder rates. This is the percentage number of patients that had at least 50% or greater reduction in their seizure frequency. And again, you can see a very nice dose response with more than 50% of subjects at the top dose exhibiting an RR50. And what was also encouraging to see that we also had on the clinical global impression scale, as well as the patient global impression scale, statistically significant improvements versus placebo for both scales. And this has to be viewed in the context of both the generation of the efficacy, but it also speaks to the drug being tolerated in the sense that the AE profile was very much tolerated in the context of that efficacy, generating these statistically significant results. So these results were very exciting for 1101, and obviously the next step for the product is to finalize the phase 3 plans and start to initiate the pivotal studies for this product opportunity. The 1101 value proposition, it's not only about efficacy, and I've shared with you and touched on the compelling efficacy data we have to date in the FOS patient population, and also the opportunity that perhaps we can actually treat depression. And this is important when you put it in the context of epilepsy patients. Up to 30% of epilepsy patients have major depressive disorder as a comorbidity. So the ability with a single mechanism, not only to get their seizures under control, but also to treat that comorbidity is an important one to reflect on when you're a clinician making your choices about which ASM you would like to utilize. But when you're developing a drug for a large indication such as focal onset seizures, it's important to have some context around ease of use. And 1101 has a tremendous profile for ease of use. It's QD dosing once a day, taken at even time with food. And the importance of taking with food is to minimize the PK variability, because 1101 does have a prominent food effect. And so to minimize the PK, which can influence both the efficacy as well as the safety profile, it's important that the patients were instructed to take with their evening meal. It doesn't require any titration, unlike the currently approved ASMs. This means we reach plasma levels with the potential to treat the seizures early in the treatment period, probably within the first week. And it's a unique and novel mechanism. So as you consider the refractory population, you're typically talking about rational polypharmacy, so having a unique mechanism rather than adding on additional ASMs with the same mechanism is a proposition that clinicians would appreciate very much. And the PK is also sufficiently forgiving that if a patient happens to miss a dose on any given day, they're not going to be at an increased risk of seizures through the day until they can take their dose again in the following day. On a safety profile, it's very much in line with other ASMs. The evening dose obviously helps with the safety profile because the highest plasma concentrations are attained during the sleep cycle of the patients. There's been no drug allergic reactions, most importantly with skin reactions, which numerous other ASMs suffer from. And there's been no pigmentation issues with 1101, which was a characteristic of the first generation Kv7 modulator, ezogabine. So I would like to thank you for your interest in Xenon today. And I would encourage you, if you'd like to know more about our pipeline and more about Xenon, to go to our website. And obviously for Bio, what we're looking forward to is meeting people in person. It's been 3 years, and to engage with our colleagues and peers, with those with a like-minded interest in the Kv7 mechanism, to discuss your potential future collaborations. Thank you.
Thank you very much. It's always great to hear what's happening in Xenon as you continue to develop this exciting innovation. So I hope everyone has enjoyed the presentations from the sampling of companies that are coming from BC. I think it's clear from what you've heard that there is some very exciting leading innovation with full of potential of making significant advances in patient care and outcomes. All the companies today will be at Bio, in addition to the other companies that are listed on the slide that is currently being presented. So please take the opportunity to connect with each other and connect with our organizations. I also want to thank our Bio San Diego supporters, whose support is critical to our ability to actually lead our delegation. And so once again, our international networking reception partner, Ferris, our partnering room supporter, Novature, and the BC government and Invest in Vancouver, who are key supporters of the delegation. They'll all be in attendance, so I hope you have the opportunity to connect with them. I'd also like to thank our annual sponsors who have been extraordinary supporters of us for many years, including the last couple of years as we've weathered our way through the pandemic. So thank you very much to them. We really appreciate all the support that you provide our organization. As we've mentioned a couple of times, we have a delegation booklet on the screen right now. Is the QR code for that. So if you click on that, you'll get an overview of both the along with the companies you heard from today, the other companies that will be in attendance. And with that, I'm going to close off this session. Thank you very much for attending. It is recorded, so if you'd like to share it with people once it's up on our YouTube channel, please do. And we look forward to seeing you in 3D next week in San Diego.